Alright, so this is the second video of the series on 3.2, which is a section on carbs, lipids, and proteins. And let me just say from the beginning that this is actually one of my favorite units. There's a lot of detail in this section about carbs, lipids, and proteins, although I think this is probably going to be the most difficult section for many of you because, one, there's quite a bit of molecular models that you have to look at. So if you're uncomfortable with that, you might have some anxiety about it. All I ask is that you push through, you try to make sense of it, and where you get lost, you make sure you ask questions, but direct questions, not open, vague questions like, can you go over that again? Because I can talk about this again and it doesn't make any sense to you, it's not going to be helpful. So you need to ask direct questions about where you get lost and what I could do to help, okay? So let's go and get started with 3.2.1, which is to distinguish between organic and inorganic compounds. In the previous podcast, I mentioned the definition of an organic compound, but I asked you to figure out if whether or not carbon dioxide, CO2, was an organic compound or not, and I hope you figured out that it is not. So let me just show you a Venn diagram, a way that I visually think about organic versus inorganic. We know that organic has to have carbon, so I'm going to draw like a diagram here that's supposed to be a circle. If you can draw a better circle than that, great. So this circle represents carbon containing compounds, and within that we have a smaller subset, and this is not showing relative size or relative amounts of compounds, but we have a smaller subset in here that represents the biologically defined organic compounds. So when we're talking about carbs, lipids, and proteins, they fit in this inner circle, whereas CO2 would fit in this outer circle. It has carbon, but is not considered an organic compound. So we're really focusing in on the things that are carbon containing, we define as organic, and you might recognize these as common nutrients that we'll talk about as we continue on. So for 3.2.2, you need to be able to identify amino acids, glucose, ribose, and fatty acids from diagrams showing their structure. This is where you need to memorize a little bit, but we will, once we finish the lab content in class, we'll do an activity in which you kind of investigate at least the amino acid structure. But that's actually where I want to start with the amino acid, and then we'll talk about glucose and ribose and fatty acids. Okay, so let me draw a generic or generalized amino acid, but I'm going to color code it as best as I can. We've got a central carbon that, then let's pick a pretty bland color here. That's got what we call, this in this whitish gray color is called a carboxyl group. You need to be able to recognize that. So this is one of the indicators that you've got an amino acid, but it's also got, I guess I should do this in yet a different color so you can know what the carboxyl group is. I'm just sorry, I'm thinking about color combos that would help you if you, especially if you have a photographic memory, which comes in very handy for this kind of thing. All right, so this is the molecular structure, and just a key point that we draw this as a model for our molecular structure because we can't see them, and this is our best representation of them. So each of these letters, if you remember from chemistry, represents an atom. When these atoms get put together in certain ways, we get certain kinds of molecules, kind of like letters and words in our alphabet. And let me just point out that this part right here we call the amino group. Now I want you to notice what kinds of atoms we have in here. We've got hydrogen and nitrogen. That's it. Hydrogen and nitrogen. Remember CHON? We've got our carbon, hydrogen, oxygen, and nitrogen in this molecule. So there are a couple of giveaways that if you were given a structure like this that you should be able to recognize. You've got an amino group with a nitrogen in it, and we've got our carboxyl group that I pointed out earlier over here, which comes in very, very significantly when we talk about putting amino acids together. But the other key that you need to know is this right here, this R. If you look at the periodic table, there is no R on the periodic table, not a lone R, and this actually represents what we call a residue group. And so there are only 20 different kinds of amino acids. Actually, I'll make these notes over here. So I'm just kind of getting ahead of myself to a future section, but there are only about 20 amino acids available in all living things, and the thing that makes them different is this residue group. They all have this basic structure at the at one end of the amino acid, and so we often say that this is the NCC backbone of one of the ends of the amino acid. So we've got NCC as our backbone, but that residue group is what makes each amino acid different, each of the 20 different. And this is, so I kind of, again, I think of this analogy of letters and words. So we only have 26 letters in the English alphabet, but we have thousands, millions, trillions of words. By putting putting letters together in different combinations, reusing them in different ways, and that's exactly what we do here when we talk about proteins, which are made up of amino acids. So we put lots of amino acids together in different combinations and different lengths. We get different kinds of proteins to give us all the different kinds of proteins that that we have. So really, it's quite important that in our diet that we ingest enough of the kinds of proteins our body needs, break them down, remember, digest them. So after ingest them, digest them, absorb them into our body so that each ribosome in each cell can assimilate them and has enough of these letters, these amino acids, to put together into the words or proteins that our body needs. Okay, so this is the structure you need to know. You can put it on a flashcard, whatever you need to do to practice this, draw it over and over, rewrite your notes, whatever it is. That's the amino acid, okay? So let's go on to glucose. So label that one as the amino acid. Now for glucose, I'm going to do this all in one color. So I guess before I draw this out, let me just tell you that what we're drawing here are the monomers, or the building blocks of different kinds of polymers or larger structures that we will continue to talk about. So let me draw the molecular structure of glucose here. Now remember that this is what we would call a two-dimensional model, but glucose doesn't exist in 2 dimensions. So these H's and OH's that are coming off these carbons are actually may not be going just up and down. They may be going forward and backwards. I hope that makes sense to you. Hold on a second here. Yeah, I messed that up. So I hope you didn't draw that. I was talking while I was drawing this. Oops, I ran out of space. I'm going to draw it real small. And then this line should be coming straight up. and that one's to the left. But what I want you to realize that for every single carbon that we have here, so we should have 6. We've got 1, 2, 3, 4, 5, 6. So we've got 6 carbons. For each carbon, we've got an H and an OH. So this carbon has an H and an OH. 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 This carbon just has an H and it's connected to another carbon atom. So what I often think about is this H, this O, and one of these H's up here. So we've got H and OH, and then this carbon over here has its H and its OH. So for every carbon, we have 2 hydrogens and 1 oxygen. So carbon, 2 hydrogens and 1 oxygen. This is really where, think of this, carbohydrate. Think of this as carbon and water. Is that what we have here, is carbon, water? Yeah. So, I hope you answered yes. So, we've got carbon and water, and so really, let me just point out that the molecular structure for monosaccharides, which this is an example of, for monosaccharides is CH2O, or C1H2O1. So for every carbon, there are 2 hydrogens and 1 oxygen. So for glucose, the formula, we counted 6 carbons, so have 12 hydrogens and 6 oxygens. A 1 to 2 to 1 ratio that comes back to our carbon water, or C1H2O1 ratio. But note that this is only for monosaccharides, and what I want you to be thinking about as we continue through this is why, oops, that's not what I want. Why only for monosaccharides? Okay? I'm going to leave that up to you to answer. All right, so that being said, we're going to draw a ribose. Now ribose will look similar. Actually, let me draw it a little lower because I've got an extension on top to draw. Okay. So if you count these up, we've got 1, 2, 3, 4, 5 carbons. So you should be able to, I'm gonna leave that blank for you to figure out, without having to count, how many are in this molecule, what the formula is. But you need to recognize this diagram as well. And one of the key components is the number of carbons. So you might actually see a slightly different structure. These are ring structures. You might see carbon and ribose as a string kind of, or chain structure. But you should be able to recognize them based on the number of carbons that exist in them. Okay? So the last thing that you need to be able to recognize is a lipid. So let me reduce this down so you can't see it anymore. Okay? And then we've got, we need to know, you need to be able to recognize a fatty acid chain. And let's draw this. Okay, we've got our OH at the end. We've got I hope you can recognize what group this is. We've got a COH double, and that C is also double bonded to an oxygen. And we've got our carbon that then continues down, sorry, keep hitting our button here, down this long chain. And this chain could be hundreds of carbons long, it could be a few carbons long. It all depends, but we're talking about large molecules here. And remember, I hope you know this, that carbon can make 4 bonds. And that's really the key to this. Why carbon's so important is because it can make these long chains of molecules. But this can keep going on, and so let's imagine this goes on forever, 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 but at the end of it, it just gets capped by hydrogen. And all along the end, the row here, we've got H's. Now, unlike glucose or ribose, you only see one OH here, and that OH is the thing that's going to contribute to a molecule being polar or nonpolar. Because this is just hydrogens, the electrons are distributed fairly evenly, so this would be considered a nonpolar molecule, therefore hydrophobic. So think back to the Pogel. So this would be hydrophobic. But this would be a fatty acid, and this is a component of fat. Okay, so you need to be able to recognize these, and we'll continue to talk about this as we go through this unit, but you need to be able to identify these from their structure. So, moving on to point 3, finally. Point 3, you need to be able to list 3 examples of monosaccharides, disaccharides, and polysaccharides. So, let's do it this way. We've got monosaccharides. I'm not going to write it all out. We've got di and we've got poly. So you know mono means 1, di means 2, poly means many. So what we're actually talking about here is molecules that have lots and lots of simple sugars to them. So let's list our monosaccharides and we'll talk about how the disaccharides are made up. So the 3 examples of monosaccharides that you should know, we've talked about one, glucose. Another one is galactose. And the third one you should know is fructose. Even though maybe ribose is a good example, all of these come back to what we're going to talk about in the future. So these would be good to commit to memory now as we continue to build our knowledge base. So disaccharides are essentially when we take 2 monosaccharides and put them together. Now these are 3 examples of monosaccharides. There are many, many more. But if we put 2 of them specifically together, we can make different kinds of disaccharides. Like putting 2 letters together, you make different words. So the examples you should know are maltose, lactose, and sucrose. Okay, and then for the polysaccharides, the ones you should know, starch, which you already know, also goes by another name of amylose, glycogen, which we've mentioned when we talk about insulin, well, blood sugar control, and it does the opposite of insulin, and cellulose. Okay, so polysaccharides are when more than 2 of these monosaccharides are put together, usually hundreds. Okay, so that's what you need to commit to memory, and then you also need to state a function of glucose, lactose, and glycogen in animals, and then fructose, sucrose, and cellulose in plants, and that's point 4. So we're going to talk about the functions of some of these. So let me just copy the charts from the textbook. So we're going to talk about animals first. Why not, right? So animals, and the examples that you need to know from the animal kingdom. So we're going to talk about glucose, and then glucose, lactose, and glycogen, the kind of saccharide that they are, and their function. So we're going to talk about glucose first, and if you look at the chart just above, you see it's a monosaccharide. So I'm not going to jot that down, but you should know this. It's in the chart. We've got lactose that we're going to talk about, and glycogen. Okay. So glucose's function is that it's chemical fuel for cell respiration. So when we're talking about chemical fuel, we're actually talking about making ATP. So remember, we get glucose into our body through the villi and microvilli of our small intestines, goes into our bloodstream takes it around to every single cell. This is the blood sugar that we're talking about. We need to maintain a certain level of blood glucose. And really, when you're starting to get hungry, it's maybe because, oftentimes it's because your blood sugar level is low. There are chemical triggers that sense that and tell you, go eat something. When you feel lightheaded, it's because your blood sugar level is low. You don't have enough glucose getting to your brain, and your brain is starting to shut down. As a preventative and fail-safe to make sure you don't lose function. You start to shut down, you faint, whatever. Your body just shuts down. All right, so lactose. So it's not really a function, but you need to know that this makes up part of the solute of milk. Think about lactose intolerance, which we'll talk a little bit more about in the future when we talk about human diet and nutrition in the next unit. But lactose intolerance, someone who cannot process lactose can't eat or drink dairy products unless it's been treated in a certain way. And again, we'll talk about that in the future. And glycogen, unlike lactose, which is a disaccharide, is a polysaccharide. And this is really, this is the polysaccharide in which humans, all animals, store glucose in the liver and in muscles. So when we have high blood sugar level, insulin takes it out of our blood, puts it into the liver and muscles, and stores it as this polysaccharide glycogen, ready to be used so that when our blood sugar level goes down, let's say you've been working out a lot, then the glucagon in your pancreas is released into the blood. It breaks down the glycogen back into the glucose molecules so that the mitochondria can make use of this chemical fuel to make lots and lots of ATP for your body. Okay? So that's really how all this is related to each other, or at least the glycogen and the glucose. But let's move on to the plants and see if we can make sense of the examples that you need to know. So let's talk about plants. And the same deal, that these are in a chart in your book, but I'm just going to copy these down and give you some context for these in case you don't know. So we've got fructose, we've got sucrose, and cellulose as the examples that you need to know the functions of. So fructose, as you see in the chart, from the chart that we just drew, that fructose is a monosaccharide, and it is the thing that makes fruit sweet. So this is the sugar that is in fruit to make it sweet, but that sugar is not meant for the plant itself. That's actually sugar is meant for us as animals to attract us because it's got lots of this monosaccharide that our body can use for cell respiration, so that we eat it and then we move the seeds that are found in that fruit to some other location, hopefully giving that seed a better chance to grow and spread to find new homes. But fructose is monosaccharide and it makes fruit sweet. That's the function as basic as you need to know. I hope you understand the larger context of that I just described. Sucrose is a disaccharide, and this is what's often transported. This is what's often transported from the leaves of plants. So in photosynthesis, we talk about glucose being made, but that glucose gets combined to make this disaccharide called sucrose that the plants then transport from their leaves to other locations through the phloem or the vascular tissues that they have that carry food. And lastly, we have cellulose, which is a polysaccharide, and this is the primary component of cell walls. This is what gives plant cells their structure, that rectangular shape that you know from ninth grade. That cellulose in the cell wall gives the plants their structure because, one, you know they don't have bones. So how can a tree stand so tall? It's because of the cellulose, which is also the molecule that makes up wood, and it also lines the xylem, which is the vascular tissue that carries water from the roots up to the leaves of a really, really tall plant. And if you look back to the drawing, cellulose is a polysaccharide made up of lots and lots of glucose molecules. Remember, glucose has lots of OHs and H's coming off of each carbon. Those OHs carry some charge, which allows water molecules to create hydrogen bonds to them as they adhere to the walls of the xylem, traveling up the plant and then leaving the leaves through transpiration. But as water leaves, the water molecules that replace them pull up other water molecules through cohesion up the xylem continuously as they adhere to the walls of the xylem. Okay, so those are the things you should also know. Let me just throw in starch, which is not on the list but is good to know. Starch is another polysaccharide, and this is the storage of energy. Just like in animals, we're talking about glycogen, plants stored as starch. We know starch very well because that's what makes up the potatoes, a lot of what's in potatoes and carrots and all those roots, vegetables, because plants store that glucose that they made in photosynthesis, transported down to the roots as sucrose, make it into starch, which are long molecules of glucose in a slightly different format than cellulose, and store it there for days like now when it's winter and plants are unable to do photosynthesis. Their leaves have fallen off. The wavelengths of light have changed, so they're not able to get the same amount of energy out of solar light, and so now they're using the reserves of starch. So when we eat their starch, we're actually robbing plants of their energy. Shame on us, right? So anyways, let's move on to, okay, so we've got point 4 down. Okay, condensation and hydrolysis reactions is what we need to talk about very quickly as possible. So let's, I'm going to draw a line here and pick a new color because this is a new idea. So we're talking about condensation reactions versus hydrolysis reactions. So condensation reactions or hydrolysis reactions. So you'll see that condensation reactions go by a couple other terms. They're also known as synthesis or dehydration reactions. And essentially these are when two monomers combine. So we're talking about molecules getting bigger, whereas hydrolysis reactions are large molecules breaking down into smaller parts. So digestion is what is a hydrolysis reaction. Assimilation is a kind of condensation reaction. So we need to talk about these in terms of monosaccharides, disaccharides, polysaccharides. Okay, when we're talking about carbs, we're talking about the saccharides. And so when we put two monosaccharides together, we get a disaccharide, and we add yet another monomer and continuously do that, we get polysaccharides. So let's see. Let me tell you this. I hope you gleaned what you needed to out of the reading. But for those of you who are still lost on this, I'm going to say see future podcast on um, condensation and hydrolysis reactions. Because I see that I'm already at 30 minutes, which is quite long. So I don't want, if you got this, I don't want to spend time and waste your time. But this is a very, very important idea that's going to be used and applied throughout this unit and future units. So this is a key podcast, but that will go up soon. Okay, so let's move on. I'm going to actually move on to 3.2.6 and talk about 3 functions of lipids. So when we talk about carbs, we're talking about sugars. When we're talking about lipids, what we're actually talking about we're talking about fats. So think about like animal fat and oils. Those are all fats. And we've got phospholipids and steroids. And the thing that they all have in common is that they are all hydrophobic, which I hope you realize means that they're all non-polar. And we have to talk about 3 functions of them. So the first 2 have to do with fats. So let me just clarify that this is fat. I'm talking about fat. So the first function of fat is that it stores energy. Now you might know carbs store energy as well, and we'll come back to this, but fats store energy more long term. Think about the long fatty acid that we talked about. Well, fats are made up of 3 fatty acids and a glycerol, and each bond between an atom is where the energy is stored. So each of those dash marks. So the more dash marks you have, the more energy that's being stored. So number 1 thing is that it stores energy. The number 2 thing is that it provides thermal insulation for organisms. So think about, so if you have more fat on your body, you're able to stay warmer without burning as much energy. You can have a low metabolism and still maintain a steady body temperature, which we often think of, well, being fat in our society, being fat is not a good thing. But if you think about our ancestors who didn't have as much food readily available to them, having fat in our diet was a very, very good thing. And that's what's led to the problems that we see today, is that we have lots of access to sugar and fat, more so than our ancestors did, but we still have that drive. It's hardwired in us to find fatty and sugary foods. And it's for these reasons. It's a good energy source. It insulates us as far as temperature goes. So if we didn't have access to as much food, these things would help keep us alive. And then the last thing actually refers simply to... Right, so I think that sorry, I had a technical difficulty there, and I lost what I was doing. So what I was talking about was the third function of lipids, I believe, and that has to do with the phospholipids. So phospholipids actually make up the cell membrane of all cells in prokaryotes and eukaryotes. So they make up the cell membrane, and if you remember, we sometimes draw them like this with phospholipid with fatty acid tails. So we've got 2 fatty acid tails, and then this part is the phosphate head, which really means, if you think about this, we know that fatty acids, based on their molecular structure, are nonpolar, which means that they are hydrophobic. And the phosphate head is actually polar, and therefore hydrophilic. And what that means is that the phospholipids actually have 2 ends to them, and the nonpolar hydrophobic part is to the inside of a cell membrane, whereas the phosphate head is to the outside. Remember that all living things live in an aqueous solution, mostly made up of water. So the phosphate head is on the outside. So actually, we have a bilayer of phospholipids along a cell membrane, and it would continue all the way down in all directions, and we would have what we call the phospholipid bilayer. Two levels, so that the fatty acids live on the inside and not touching water, and the outside of the cell membrane touches water on either side of the cell membrane. Okay, and those are the three functions of the lipids. Excuse me. And the last thing you need to know is you have to be able to do is compare the use of carbs and lipids in energy storage. Well, carbs are smaller molecules, so they're used a little easier than lipids and a little faster than lipids. But per gram, fat stores twice the energy that carbs do. So think about why it's so much harder to burn off fat. In order to burn off fat, you actually have to run through all of your carbs first, and then your fat. But fat stores twice as much energy. So to burn through, you have to burn through twice as much energy. So it makes it that much more difficult. But fat for the long haul is good, because if you don't have a food source, fat is really good to already have in your body, to continuously use it as an energy source. Okay? That's long enough. I hope it makes sense. Jot down any questions you have, and make sure you ask them to me, okay? All right, this will finish out 3.2.